what's I would say is colloquially known as salvia, and this might be a conversation you will have after this lecture. You heard about salvia? Isn't that a mint plant? No way! So, have you heard of Salvia divinorum? That's what I'm going to talk about. So, Salvia is the genus, so this name here, and then divinorum is the species. So it's a rare species that actually was only found naturally in a geographic region of Mexico, and the native peoples there used to use it for religious rites and things like that. So the actual active compound that you would be taking, like in a clinical study or something like that, is salvinorin A. So that's the component of the plant. So that's what it looks like here. It's kind of overwhelming in my opinion. So that looks crazy. It must be a terpenoid. Yeah, so for some of you, actually, I think a good portion of you have probably taken natural products with Bruce Greenberg. Anyone? Yeah. So, made up of small 5-carbon units. These two are the initial precursors to all terpenoids. And they combine together to make like very large compounds. So you have things like cholesterol and then like tons of other natural products. So this is just also from his notes. You probably all recognize this. Just the initial synthesis here. So you have DMAP and IPP, and then they combine to make a monoterpenoid. Now, salvinorin A is a diterpenoid. So if you attach another, I believe it's DMAPP. No, it's IPP. If you attach another IPP, it can fold in such a way and be altered so that it will become salvinorin A. So here's some other.
Sam, you're studying from the slides.
Oh, sorry. Sorry. All right. So we have menthol. It's another terpenoid. Estrogen, hormone, cholesterol. These are all terpenoids. So usually they're produced in plants, used for things like self-defense. So insecticides, bactericides, fungicides. There's actually some cases where some plants produce them and it will actually harm things that commonly eat it. And so like I said before, with estrogen, hormones, and then tons of other natural products. So the reason why I want to talk about this is because salvinorin A is quite unique. In the respect, like I'll get into it further, but the fact that it's a terpenoid and that it has the actions that it has makes it very interesting in terms of research perspective. So there aren't many studies done on the physiological effects of salvinorin A. I did find one that they tested it on rats. They dosed them with a wide variety of concentrations, and then basically I think it was for a 2-week period, and then at the end sacrificed them and examined the histopathologies of these organs, and they found no significant histological changes over a 2-week exposure period, even though they administered doses of up to this concentration in the rats. So they concluded that it had very low to no toxicity in that respect. But on the mind, we got tons. Tons of effects. So the main mechanism in salvinorin A is on the kappa opioid receptor in the brain. So you have a few types of opioid receptors. You have 3 main ones. There's the mu, the delta, and the kappa. And I'll get into that more later. So again, this is another special part about salvinorin A being a terpenoid and all that stuff. It's the only known non-nitrogenous compound to affect this receptor. So I guess that just means like no amino group or anything like that, which is very unique. So, opioid what? Basically, it's an opioid receptor, as I said before. So the other 2 main ones are the mu and the delta. So those ones generally are responsible for anesthesia and like the reward reward response. And up until salvinorin A, actually, well, it should say around that time, but salvinorin A was kind of revolutionary in that they had labeled the kappa receptor as dysphoria, or like you'd feel ill or in an off mood. It wouldn't generally be associated with the effects that it is. So yeah, there's the kappa receptor. These are opioid receptors throughout your body, so they're in your digestive tract, up here in your brain. And so this is just kind of a generalized diagram. So you take the anesthesia, it gets digested, it goes to your brain, and then something goes down there to numb the pain, the metabolite, or the signal that it's conveyed. So there are many opioids, but the most common ones that probably would think of if you hear like opiates, so morphine, codeine, methadone, and then ones that are produced in your body like endorphins. So if you have like a pain response, then endorphins are usually produced in response to that. So opioid receptor activation has been linked to these things. So pain relief, respiratory depression, euphoria, hormone release, inhibition of digestion, and it's been known to have effects on anxiety. And then, like I said before, usually the kappa receptor would be responsible for dysphoria. So, how do they work? Morphine has been found to work on the mu opioid receptors, which I guess a lot of them are present in the brain, and so they found that when these receptors are activated, it's also in the same area that a lot of noradrenaline receptors are activated, and so they think that they act kind of together. And from the reading that I did, it didn't get into a lot of general aspects to what opioid receptors, but for the most part, this is what they would say, is that it would stimulate like reward responses. So for things like eating and other things like that, essential activities. So salvinorin A, it stimulates the kappa 1 and the kappa 2 opioid receptors, which, like with Ryan's cannabinoids, it inhibits adenylate cyclases, and then it inhibits the calcium channels, and then it also activates potassium channel influx. But it doesn't work on the 5-HT receptor, which I haven't talked about, but if you've heard of it before, it is one that's generally responsible for hallucinogenic effects in your brain. And so that's another reason why this compound is really unique, because it doesn't affect this receptor, yet it has those effects. So, before scientists found it, it was used by these people in a specific region of Mexico. And yeah, so I don't know how this got involved, but that's what they think. And then yeah, and then teen hippies have now joined the spiritual party. All right, so I guess a lot of what I learned from doing this research was just what we know about salvia and what we don't know about salvia. So up until that point, or around that point, this receptor was for a completely different purpose. It is now said on Wikipedia, no less, that it causes dissociation responses, and as well as hard to, I can't recall the exact word, but basically a distortion of your reality. And so this receptor had not really been thought of responsible for that before. And so it was pretty interesting for a lot of people. It seems there's been a lot of research done. And so yeah, deliriums, that's the word. So yeah, and then I think that kind of opened the door for me thinking that lots more research should be done on this area because, you know, firstly, it's a diterpenoid. no nitrogen, doesn't affect the 5-HT, and yet it's responsible for these deliriant and hallucinogenic responses. And yeah, it does a lot of other things.